Okay, thank you. Well, good afternoon. I'm very happy to be here. Thank you very much for inviting me, and thank all of you for showing up for this talk. And I hope it will be worth your while. And I'm hoping to learn a lot from you too, because I understand a lot of you are specialists in some of the things I'm going to be touching on. My knowledge of these things is very thin, okay? And I think you go deeper than I do on these various subjects, so we'll see how it goes.
I just want to say that I talked to Helene Nytorp, and I want you to stand up here. She's the person who has been the driving person that you are here.
Okay, well, thanks again, Helen. Okay. All right. Well, I will be talking today on myalgic encephalomyelitis and chronic fatigue syndrome. And that is my name. It's a Dutch name. It means from Rabbit's Mountain or Rabbit's Town. But you can just call me Rich, okay? Everybody, please, just Rich is good enough. So here we go. This is what I hope to cover this afternoon and also some tomorrow morning. And I don't know how much all of you already know about chronic fatigue syndrome or myalgic encephalomyelitis, so I'm going to kind of go through some of the basics about that, and if it's boring, just start snoring and I'll know that I should go faster. So we'll talk about the history. See, there we go. The history. We'll talk about the definition of these disorders, the symptoms, who has the disease with the epidemiology, the genetics, and then the likely causes. And then we'll get into some biochemistry to prepare for talking about this hypothesis that I have. So we'll talk about glutathione, the methylation cycle, and the whole sulfur metabolism. And then we'll get into this hypothesis, and then how it explains all these various symptoms and so on from chronic fatigue syndrome, how it matches up. And then eventually we'll get down to how you can test to find out if in a given case, if this theory applies. And then, oops, sorry, wrong button here. And then we'll talk about treatment based on this hypothesis. And then I'll talk about, at the end, a clinical study that we did using this treatment and what the results of that were. Now, I put a lot of information on my slides, and it's probably too much, but the purpose of that is that after I'm dead and gone, if you still want to learn more about it, you can get a copy of this and you can read it, and it should be self-explanatory. You won't need me, okay? So these are not notes to me. These are pretty much the whole talk here. I do want to mention a couple of disclaimers. First of all, we'll be talking about treatment later on, and anybody who undergoes this treatment should be under the care of a licensed physician. I'm not a licensed physician. I will be talking a lot about hypothesis, and I'll try to make it clear which things are my hypothesis and which things are accepted as scientific fact today. And if I don't do that, raise your hand and say, do you have any proof for that, or is that your fairy tale, okay? So I'll try to keep that clear. And I didn't put a lot of references in this talk, but I have papers on the internet, and most of the references are in there. So I didn't put a lot of references here. And then finally, I'm not trying to sell you anything except ideas, okay? I don't sell supplements, I don't sell testing or anything like that. So. All right, so start out with the history. And first I'm going to talk about the history of myalgic encephalomyelitis, and then later the history of chronic fatigue syndrome, and then we'll talk about them together. It was originally defined by Dr. Ramsay in the UK in 1956. And he did that based on the experience of an epidemic at the Royal Free Hospital that happened in 1955. The definition, strictly speaking, means pain in the muscles and inflammation in the brain and the spinal cord. Now, for many years, there was a debate about, is there really inflammation or not? But in recent times now, there have actually been some autopsies on people who died who had ME, and sure enough, they did find, I've got to learn how to push these buttons here. They did find inflammation in the dorsal root of the spinal cord. So apparently this this really isn't an accurate description, at least of some of the cases. There had been previous epidemics, and one was in Los Angeles in 1934, but there were also many before that. And Dr. Byron Hyde, who I think has visited Sweden not too long ago, maybe a year and a half ago, he has done a study of the history of ME, and he traced it back to the ancient Egyptians in 1900 BC. So this is not a new disease. It just wasn't recognized, wasn't named, and so on. But
when they started to eat grain.
Is that what did it? Okay, well, there you are. So problem is solved. Okay? Now, all right, now, what about CFS, chronic fatigue syndrome? What's the history of that? Well, that was first defined by the Centers for Disease Control, CDC, in the U.S. in 1989. And it was a response to clusters of cases or epidemics that occurred in a few locations, and the main one being near Lake Tahoe. And again, it looks as though this had existed for many years under different names. It seems like it's more prevalent now, starting in the 1980s, but nobody can really prove that because we don't have good epidemiology before that. And again, the cause and causes have not been established and agreed upon. Now, I'm going to suggest what I think the causes are, but the community as a whole is still arguing about that, okay? And the most recent thing is the XMRV retrovirus, and maybe you've seen in the news recently, now they're saying, oh no, it's not there, you know, so, but the people who originally identified it are not giving up yet. They say, oh yes, it is there, and they're doing some additional testing now and using different analytical techniques to try to make sure that they know what they're doing. So that one I'm not ready to say one way or the other, but I think it's quite possible that it's there based on the fact that I think the people who did the work did good work, and also there are physicians treating as though there is a retrovirus, and they're seeing some positive response, even though they can't say for sure that it's there. So, you know, I think it's a good possibility. Now, as you may know, in the UK and some other countries like the Netherlands and Belgium, chronic fatigue syndrome has been taken over by the psychiatrists. And this is not necessarily a good development from the standpoint of the people who have this disorder, because it's really a physiological disorder, not a psychiatric condition, although people do experience depression and things like that as part of it, but fundamentally there's a physiological issue. So what's been happening now is there's been a controversy as to whether CFS is really the same thing as ME or if they're two separate conditions. But the trend in recent years has been to put them together into ME-CFS, and that's like the title of this talk, ME-CFS. And most recently, the people who believe in a physiological basis are trying to get rid of the CFS part and just call it all ME. Because chronic fatigue syndrome is not a very serious sounding name. It's kind of trivial, and a lot of people make jokes about it, and the people who have it are very unhappy with that name. So it looks like we're going to be moving away from chronic fatigue syndrome, leaving that to the psychiatrists and moving on to ME. And that's probably what's going to be happening. Okay, so definitions. There's no agreed upon diagnostic test, and that makes it very difficult to define. So it's been sort of a process of elimination. You rule out other things that might cause some of those symptoms. If none of those are there, then you move on to chronic fatigue syndrome. And that's not a very satisfactory way to define a disorder, but that's what's been done. So the definitions have been developed by consensus of committees. People get together and they talk it over and they decide what's going to be the definition. And they've attempted to identify a complex of symptoms that's specific to ME-CFS or ME-CFS together to use for diagnosis and also for research purposes. So these definitions do not include all of the symptoms. They try to include those that are most characteristic, so that you can use that for a diagnosis and say, this is different from these other disorders. So that's why you just see certain symptoms. And again, this has been very controversial, and it still is. So here's a list of definitions up to the present. This just a few months ago they came out with a international consensus criteria. But prior to that, there's the Ramsey definition that came from the 50s, but it was updated. So I've got it, 1986 is when they sort of finished modifying that. And that was for ME. This was the first chronic fatigue syndrome definition by the CDC. Then this was done in Australia. And then the psychiatrists in the UK developed the Oxford criteria. Then the CDC modified the Holmes criteria, and that was called the Fukuda criteria. And that one is probably still most used in research papers. If they select patients, they say, we selected them according to the Fukuda criteria. Well, but since then, there's been other action here. Bruce Carruthers and a group in Canada put together a clinical working definition. They had clinicians come from several countries to talk it over, and they came up with that one. And then the CDC, under Reeves, sort of modified the Fukuda criteria, and they tried to quantify the measurement of symptoms. And a lot of people are very unhappy with the results of that. Mr. Reeves is no longer in charge. He's been moved to another position after objections from a lot of people. But we still have that empirical case definition. And so then there's been some more. Leonard Jason, who's an epidemiologist, has developed one for children, pediatric case definition. And then finally, Bruce Carruthers and another group, some of those were the same people, and then there were some new people, put out a new one this year. So I'll be talking about this most recent one because I think as time has gone by, these definitions have improved, and the later ones are better than the earlier ones. But you can probably get controversy about that too. So here's that's the reference for this this new criteria, and most recent. And there were 25 people involved from 12 countries to develop these criteria. And they don't use the word chronic fatigue syndrome anymore. They talk about myalgic encephalomyelitis, ME. Okay, they're trying to get rid of that term. So here are the requirements. And this first one here, post-exertional neuroimmune exhaustion, is PENE. They call it PENI, okay? PENI, that's the abbreviation. Well, this they believe is the most characteristic feature of chronic fatigue syndrome. There's fatigue present in many disorders, but if the person has exercise one day, the next day they're in worse condition, okay? Post-exertionally, they're more exhausted the next day after they've had exercise. And that seems to be a characteristic of this disorder that you don't usually see in other disorders. So that's kind of become the main be-all and end-all. You have to have that one, it's required. And then there a combination here of a symptom from neurological categories, a combination of immune, gastrointestinal, and genitourinary impairment, and then energy metabolism transport impairment. So it's sort of a combination of several of these have to be present. All right, now this kind of elaborates a little more on those things. And the full text of the criteria goes into even more detail about each one of these. So if you really want to boil it down to what actually is looked at in the clinic, then the final document here is more informative than what I have. But anyway, this first one, the post-exertional neuroimmune exhaustion, a lot of big words here, but basically it means that after a person has had exercise, they have even less energy and really can't function very well. And it's not just physical fatigue, but also involving the brain and also the immune system. So a lot of things get worse for this person after exercise. Neurological, well, there's cognitive stuff, pain, sleep disturbance, sensory, perceptual, motor, the whole range of things involving the brain. And then the immune system and the gastrointestinal, they're flu-like symptoms. They're susceptible to viral infections, nausea, a lot of gut problems here, bloating, urinary urgency, usually a high urine volume every day.
Excuse me, what is bloating? I don't know.
Pardon?
What is bloating?
Bloating. Oh, your gut gets very big. Gas. You have a lot of gas, and yeah, very uncomfortable. Okay. This is having to get up in the middle of the night to urinate. Food sensitivities, sensitivities to medications, odors, and chemicals. And then here we have problems with the heart, cardiovascular, respiratory, problems with controlling the temperature, and being intolerant of high or low temperature. So, as I say, if you look at the full criteria, it's explained in even more detail. But you can see there's quite a range of symptoms. So here are some other aspects. What about fibromyalgia? Well, a lot of people satisfy the criteria for both fibromyalgia and chronic fatigue syndrome. The first definition involved pretty much pain and pain sensitivity, and they had tender points. You push on the body at certain spots, and if the person experiences pain, that's a tender point. Well, most recently, they've expanded this definition, so now it includes several other things. Categorical scales for cognitive symptoms, unrefreshed sleep, fatigue, and some number of somatic symptoms. So the definitions is starting to look more like chronic fatigue syndrome. Fibromyalgia is sort of coming together with chronic fatigue syndrome. Is it sudden or gradual? Well, there are some cases of each. There are some sudden cases, and then there's some where people get ill after many years. Epidemics or clusters, or sporadically? Well, there's some of both. There have been clusters, and then there are isolated cases where someone all by themselves gets sick. Severity, that ranges all the way from somebody who's still able to work full-time to somebody who's in bed full-time, and some people die, okay? And there's a whole range of severity. Sometimes the situation gets worse with time. Usually it does. So anyway, those are sort of the general details of it. Now, symptoms. As I said, the symptoms in the definition are only chosen to be very characteristic, but there are lots of other ones. I've seen lists of 60 or 70 symptoms that various people have reported who have chronic fatigue syndrome. Now, this is a study from Germany in 1996, and this is a list of the symptoms that these patients mentioned in the order in which they're most mentioned. So the one they mention most here is fatigue and exhaustion, but then there are brain-related things, sleep problems, pain in the muscles, mood problems, headache, respiratory, depression, and it goes on down. You can see quite a range, gastrointestinal things here, nervous system things, immune system things. It just goes on and on. There's just a huge range. And you can imagine when these people go to the doctor and they say, what's your symptom? And they have 60 or whatever, you know, it's just bewildering. I mean, what do you do now?
And the doctor doesn't understand that the mitochondria are not functioning.
That's right, yeah. We're going to talk about that some more here. Okay, so how do we explain such a variety of symptoms? It affects so many organs, so many body systems. How do we explain that? Well, when I first started thinking about this, it seemed to me that there must be a problem in the basic biochemistry that can affect many cells. How else can you affect so many parts of the body at the same time? So then I thought, well, what are the important parameters in biochemistry? And these are the ones that occurred to me: temperature, pH, concentrations of various substances, and the redox environment in the cell. And so then I thought about, well, which one is the most abnormal? I mean, there are some changes in all of these in chronic fatigue syndrome, but which one seems to be the worst? And it seemed to me that it was the redox environment. In 1993, Dr. Ali, who's in US, published a paper where he hypothesized that chronic fatigue involves accelerated oxidative molecular injury. I think he was the first one to talk about oxidative stress in chronic fatigue syndrome. Well, by now, there are probably more than 20 studies showing oxidative stress being a problem. And that means that the redox environment has gone more oxidizing than it's supposed to be. And so then I asked myself, well, what could be wrong there? What controls the redox environment? And mainly, it's glutathione. Okay, this is the first thing that started to get me interested in glutathione as a possible issue in chronic fatigue syndrome. So hold that thought. We're going to come back to glutathione again here. Well, what about genetics? Is there a genetic predisposition in chronic fatigue syndrome? And yes, at least for the sporadic cases. Now, the epidemics, I'm not so sure. If you're in a room of people and nearly everyone in the room gets sick, that doesn't sound like a genetic issue. But when you're talking sporadic cases, there is good evidence for a genetic component. And you notice this one right here, that was done in Sweden, okay? And then there were several others there. Oh, let's see, I pushed the wrong button again. There we go. But they range from studies in families where they look at whether the chronic fatigue syndrome occurs in a first-degree relative and a second-degree relative. And the most recent one there, done in Utah, gives pretty good evidence that there's a prevalence in families that's higher than normal. So
You also learn what to eat in families.
well, that's right.
Epigenetic things must be very important here. We have no control of that.
Well, that's right. But they did look at more distant relatives, which helps a little bit. But yeah, I mean, if they're all living in the same house, they're all eating the same food, yeah, you got to wonder about that. But they also saw it moving out into more distant relatives.
They bring habits with them.
Yeah, well, too. But that's not the only source of evidence, okay? There are also the twin studies, and there are also measurements of polymorphism frequencies, and there have been quite a few. And I was at the meeting in Ottawa a few days ago, last weekend, and there are some more not on my list yet that were reported there. So there's some more polymorphism. So the collection of evidence here, I think, does say that there's a genetic component. We don't necessarily understand all aspects of it, but I think we can say that it's there. Well, here are the ones that have been reported, the polymorphisms that have been found more frequently in people with chronic fatigue syndrome than in the general population. And of course, the whole genome has not been looked at, okay? These are the ones that have been looked at and found to be more prevalent. But they involve the immune system, neurotransmitters, the HPA axis, and then here's some additional ones that are just recently reported. And at Ottawa, there were several more of the cytokines, interleukins, and so on reported again, in addition, that I don't have on this chart. So anyway, there are quite a few that do seem to be more abundant in people with chronic fatigue syndrome. So more evidence for the genetic component. Okay, now what about other ones? And besides the one I just showed, and yes, there are other ones, and this is where Dr. Amy Yasko comes into the picture. She has focused on the methylation cycle and related pathways, particularly in people with autism. But she also applied somewhat to CFS, and when I learned about this, I began to emphasize that more and got in contact with her. And as I told her, I'm going to send a tidal wave of people towards you, and I did. She was kind of overwhelmed with all the chronic fatigue people. She already had a lot of autism people that she was trying to help. But she's been very helpful to us. Now, her work has not been documented or replicated in peer-reviewed publications. Dr. Yasko's view is there are too many children who are ill and have autism, and we need to help them, and I'm going to spend my time helping them rather than publishing and arguing with the editors and reviewers. Now, there are pros and cons with that approach, okay, but she has right now 11,000 people on her internet group, mostly parents of autistic children, and she's helping a large number of them. But, you know, if you're a scientist, she's a scientist too, of course, but if you're really focusing on the science, you want to know, you know, we want to see some replication, you want to see some peer-reviewed publications, you want to know, is this the real story or what, okay? We don't quite have that. And so what I'm saying here is there's a need for good statistical analysis to really see what's there and is it a cause. I personally think it would also be helpful if there would be a search for polymorphisms in the glutathione system and the antioxidant system in general, because in my model, those are the first things affected. And in order for a polymorphism to be important, I think it has to be present in enzymes that are important early in the pathogenesis. Otherwise, it won't be accessed. So it has to be early, and I think this is the early step. So I think eventually we'll see some more polymorphisms there. Okay, now the epidemiology. Come on in. When you don't have a good definition that everyone agrees upon, then you're going to get different values for prevalence. And that's why you see such wide numbers, a factor of 10 difference here in the various studies. But anyway, these are the numbers that have been reported. In the US, this is Brigitte Evengard in the Swedish Twin Registry, and she looked at CFS-like illness. In other words, it had the symptoms of CFS, but they didn't bring all those people in, have them examined by a physician and decide for sure if they had it. They just did it on the basis of telephone surveys. So this number would probably be kind of high compared to what it would if you had a strict definition for chronic. But anyway, it gives you a number, and it's not too different from the biggest number here. And then just this year, 3 regions in England, and you can see these are smaller numbers. Well, it depends on the definition they're using. They're getting tighter definitions as we learn more about this, and as you get a tighter definition, you have a smaller prevalence number. So probably these lower numbers are better than these higher numbers. And of course, we can't say for sure it's the same in every country, but who knows? I mean, maybe it is, maybe it isn't. But my suspicion is that the American numbers are a little bit high because the definition was not as tight. Here's a Japanese one this year, and they have 10 per 1,000 people. And so those are the kind of numbers that we have. And it's estimated that there are at least several million people in the world who have chronic fatigue syndrome. It's higher in women than in men, and I think that all the studies pretty much agree with that. They don't necessarily agree on the number, but it is higher in women than men. People argue, of course, the usual things about, well, maybe women go to the doctor more often, maybe they're more willing to complain about their problems and so on. I don't believe that's true.
Is the inheritance through the egg or the sperm?
I don't know the answer to that.
Because the hypothyroidism is coming via the egg.
Uh-huh. Well, I do know that there is quite a lot of prevalence of, say, a mother who has chronic fatigue syndrome and a child who has autism. But I don't know that that's really been studied very precisely yet.
What's the 6-month prevalence even going? What do you mean with the 6-month prevalence?
Okay. What I meant to say there was they've had the symptoms of a CFS-like illness for at least 6 months. That's what I meant to say, sorry. Yeah, in the early definitions of chronic fatigue syndrome, that was one of the requirements. They had to be sick for at least 6 months because a lot of people have fatigue, you get the flu or whatever, you have fatigue, but it goes away, But if you've been fatigued for 6 months, then you've got to start wondering about chronic fatigue syndrome. Okay, adults is higher than in children. Yeah.
Prevalence is higher in women. Does that apply to all ages?
I don't know if that's really been studied. Usually, the sort of average age at which a person gets chronic fatigue syndrome is around 30 years old. And then they tend to remain ill the rest of their life, more or less. Okay, so I don't know, though, specifically about the age groups. Okay, minorities, at least in the U.S., minorities have higher prevalences. We don't know why. And estimated that only about 15% of those who actually have it have been diagnosed. A lot of the doctors don't diagnose chronic fatigue syndrome. They don't recognize it or they don't understand it, and so and a lot of the people probably in the minorities.
If there are no pills,
Yeah.
then they will not know.
All right. I think you got a point there. All right. All right. Now, what about the causes? Well, now I'm going to get into how it looks to me. All right. So we're talking now some hypothesis here, but there have been risk factor studies where people have interviewed those with chronic fatigue syndrome or given them questionnaires and say, you know, give me some history, what happened to you before you had the onset of this illness? And when they do that, they find these sorts of things, okay, and quite a variety. I mean, every person you talk to has a different story and different things happen. But there is a common denominator in the sense that they're all what I would call stressors, okay? Stressors in the sense that your body's stress response system responds to them in a common way, even though they're different. So physical, chemical, biological, emotional, psychological. And the physical can be things like a trauma. It can be too much exercise, like running a lot of marathons, too many marathons. It can be surgery. The chemical can be a variety of toxins, pesticides, solvents, ciguatoxin, like from fish.
And amalgams.
Pardon?
And amalgams.
Well, yeah, exactly, and I'll get to that later. But the available risk factor studies have not really said that, okay? I agree with you, but the studies don't mention it, which is unfortunate, you know? I think people have kind of steered away from that. They don't want to talk about it, but it's there. Okay, biological, infections, immunization. Now, this is a big controversy, you know, in autism as well as chronic fatigue syndrome. But there are a lot of people who do report that they became ill after an immunization. Blood transfusions, insect bites, and of course there you're starting to get into Lyme disease as well, because tick bites can produce Lyme disease. Allergic reactions, and then just not eating or sleeping properly, and we have a lot of that. Well, then emotional and psychological, and I have another slide that talks about it. But I want to emphasize here, this has been a very controversial thing because some people conclude when they see this that, oh, it's a psychiatric illness. Well, I'm not talking about that, okay? I'm talking about how these sorts of stressors impact the body's stress response system, and then that has a physiological effect. So let's see what that, okay, well, I'm sure that none of these things have ever happened to anyone here, okay? But some people do suffer some pretty bad things in their life, okay? Someone dies in the family. Let's see. Got to learn how to handle these buttons here, or not handle them. You know, deaths, even getting married, if it's a good marriage, is stressful. If it's not a good marriage, well, of course, that's stressful, you know. It's hard to win the situation. Hopefully, in between, it's good, you know. Separation, divorce, illness, job loss, new job, moving from one house to another. Difficulties, well, yeah, difficulties. We all probably have some experience with difficulties. And, you know, too much activity or busyness, overwork, you know, the workaholic kind of a person. We have a lot of that kind of thing. Dilemmas, trying to decide what to do, and both options are bad, and if you don't do anything, it's going to get worse too, and you're stuck, you know, that's a dilemma. And then the childhood problems. There's been another study out of CDC on this one. Unfortunate things happen to children in families and whatnot, and those have been correlated with developing chronic fatigue syndrome also. Okay, so how do we pull all those stressors, all those potential causes together? How can we make sense out of this? And well, all of them, in accordance with the body's non-specific stress response system, all of those cause increase in the secretion of cortisol, epinephrine, norepinephrine. That's the body's non-specific response. Now, the body has specific responses to all these stressors also. If you're cold, you shiver. If you're hot, you sweat. You know, there are specific responses. But in addition to the specific ones, there are the non-specific, and in that case, they're all treated together. Apples, oranges, bananas, everything, all in one collection, okay? They're all, the body responds to the whole thing together. Well, so this happens initially, and notice I say initially, because especially I know some of you are interested in cortisol, and that tends to drop later on. But initially in this disorder, it goes up in response to stress. Well, ultimately though, directly or indirectly, all these stressors end up placing demands on good old glutathione. Okay, that's the bottom line, and that's where they come together. They may be very different stories for each person, but if you look at the whole story, they're loading up their glutathione and it's going down. Wolf Droge and Holm, they were in Heidelberg, University of Heidelberg. They first suggested in 1997 that glutathione is depleted in this disorder. And as far as I know, they were the first ones who suggested that. And then Paul Cheney in the US, who was present at the Lake Tahoe outbreak of chronic fatigue syndrome in the 80s, he reported that nearly all of his patients had depletion of glutathione. So this got me very interested in glutathione, and I started studying glutathione. And before that, I'd never heard of it. Okay? And today, just as you said, there's no pill. Most people have no clue. What is glutathione? You know, so we're going to talk about glutathione. Here it is, okay? So
there is an interesting story about healthy centenarians in the U.S. They are high in glutathione.
yeah, I've heard that. It's sort of a measure of how long you're going to live. So, and some people, you know, devour glutathione for that reason. So anyway, here it is, and it's a tripeptide. It's like a very short protein. It's made up of three amino acids, and the important thing here is notice that it has a sulfur group. It has a sulfhydryl group there, and that's the business end of this molecule. Okay, it's able to bind with toxins. It's able to bind with another glutathione molecule to form an oxidized glutathione. But that's the important part, that sulfur there. So again, it's a tripeptide, and these are the amino acids. The cysteine is the one that has the sulfur in it, found in all the cells, in the blood, in the bile, and the lining fluid in the lung. So this thing is present pretty much everywhere in your body. It's made mainly by liver cells, but also red blood cells. They're the main suppliers of glutathione to other cells in the body. It's the most important thiol, or sulfhydryl containing substance in the cells. It's the highest concentration. And as I said, it has reduced and oxidized forms. Two glutathiones can get together, and then it forms a disulfide bond, and it's called GSSG then. The ratio of glutathione to the oxidized glutathione is what controls the redox potential in the cells. So this is the important aspect inside the cell. And it also serves as a basis for the antioxidant system, which quenches reactive oxygen species. And here's some more things. It conjugates several classes of toxins, and one of the favorite ones here is mercury. Well, it binds mercury. It's one of the toxins that glutathione takes out. So it's responsible for phase 2, one of the pathways in phase 2 detox, but it also supports phase 1 detox in general. So when glutathione goes down, it's not just the toxins that it usually binds, but all the toxins really that are influenced by phase 1. It supports the immune system, especially the cell-mediated part of the immune system. It plays an important role in making proteins that contain cysteine, and I'll talk more about that. It participates in making bile. This is an important one. It protects vitamin B12 inside the cells and also assists in the metabolism of B12, and we'll be talking more about that one. And of course, it has many other things. So glutathione is just a wonderful substance. The more of it you have, the longer you're going to live, and unfortunately, these folks don't have enough. All right, now I want to talk about, hey, you got your jacket back, great. He left his jacket on the train. All right, now this is just a brief sketch of some biochemistry, and I'll show you some more detailed slides later on here. But I just want to point out this red one here is the methylation cycle. The green one is the folate cycle, and they are linked together right here in methionine synthase. That's an enzyme. And then this blue is the transsulfuration pathway. Here's glutathione, And then it goes on down, the sulfur metabolism goes on down to make taurine and sulfate, and then those are the main excretion forms of sulfur. So this basically represents the sulfur metabolism in general. And as you can see, it's fed by our diet, eating protein. Protein supplies methionine. It also supplies some cysteine. Now, our normal diets don't supply enough cysteine for the demands of the body, so it's about half. It supplies about half what we need. So the other half is made from methionine by this pathway here. So methionine goes around to homocysteine, and then that goes down and makes cysteine. And so now I'm going to show you a more detailed sketch, but when I do, just keep in mind the red is the methylation cycle, green is the folate, and the blue is the transsulfuration. Okay, I keep the same colors here. We'll see that in a minute. So what does the methylation cycle do? Well, it's present in all the cells of the body. It makes methyl groups, CH3, and those are used by a large number of biochemical reactions in the body. Probably 50 or more different reactions need methyl groups. So this is a little factory for making methyl groups to supply to other reactions. It also controls the overall sulfur metabolism. It balances the needs for methyl groups and for glutathione to control oxidative stress, and then also the other sulfur metabolites. So that methylation cycle is very important, being at the beginning of the sulfur metabolism. It's in a place where it controls the whole thing. And when something goes wrong there, it affects everything after that in the sulfur metabolism. And together with the folate metabolism, it coordinates the production of new DNA with the supply of methyl groups, which are used to methylate DNA, among other roles.
Is that what is behind that methylation of DNA, behind the expression of genes?
Mm-hmm, that's right.
Epigenetics?
Exactly, yeah. When DNA is methylated, when methyl groups are attached to it, it silences the gene, so it's not expressed. And so methylation is very important in controlling the gene expression. And folate is important in making DNA because folate is needed to make new DNA. So by coordinating the fact that these are hooked together, the methylation cycle is hooked to the folate cycle, that can be coordinated. So you do enough methylation of the DNA that you do make. And when one goes down, then the other goes down to try to control that. Because it's important to have the right degree of methylation. Well, okay, B12. I'm going to be talking about that in the next slide. So what are the functions of vitamin B12? Well, there are two. One is when it's in the form of methylcobalamin, or methyl B12. It acts as a coenzyme for this same enzyme we've been talking about, methionine synthase, which links the methylation cycle and the folate cycle. And that enzyme converts homocysteine to methionine. And the other form, adenosyl B12, adenosylcobalamin, acts as an enzyme for a coenzyme for this enzyme, and it helps to feed fuels into the Krebs cycle, which helps to make ATP, and that's the energy source for a lot of reactions. So those are the two things that B12 does in the human body. What about folates? Well, folates act as a source of methyl groups for the methylation cycle. The methyl group originates in the folate metabolism, and then it's passed over to the methylation cycle, makes S-adenosyl methionine, or SAMe, and then is able to be supplied to these 50 or so reactions. So the methyl groups actually start in the folate cycle, and then they're passed on from the methylation cycle. Folate also participates in the metabolism of histidine, which is one of the amino acids. It also synthesizes thymidine, which is where it's needed for making DNA, and also purines, which are needed for DNA, RNA, and ATP. So folate has a lot of very important roles here too. All right, so now we get a little more complicated, but remember the red one, this is the methylation cycle, this is the folate cycle, and then we're going to talk about transsulfuration later. But right here, MTR, that's that same enzyme, methionine synthase. And that's one that I hope you'll remember because that's the key to this whole thing. Okay, that enzyme is partially blocked in chronic fatigue syndrome. That's the key to the thing. And that ends up affecting both the methylation cycle and the folate metabolism. And it ends up affecting everything down below here too. So that right there is the key. Now notice I've got some of these in bold. B12, as I mentioned earlier, is a coenzyme that supports that enzyme. And then there's a form of folate here, 5-methyl tetrahydrofolate or methylfolate, which is a reactant which is used by this reaction. And then you can see it forms tetrahydrofolate. So this one loses its methyl group and becomes tetrahydrofolate. It gives its methyl group to homocysteine, and that produces methionine. So basically it just passed on a methyl group. And B12 helps it do that. Well, the rest of this is interesting, I guess, but I don't know if I'll have time to talk about all of it. But this reaction right here, betaine homocysteine methyltransferase, this reaction is present only in the liver and the kidneys, not in other cell types. And if you take betaine or trimethylglycine, that feeds that reaction, and it's another route for converting homocysteine into methionine. So it's in parallel with this methionine synthase, which is the one we're interested in. This one is blocked, partially blocked. Sometimes it's beneficial to get this one going a little bit in order to make S-adenosyl methionine, because that's needed to regenerate this one, okay? So this thing is all kind of like a spider web. But anyway, I think we've got the main ideas for here. Let's move on. Now, this is the transsulfuration pathway. This is just below the part I showed you. So there's homocysteine, and now we have the blue line going down. And so you notice it goes through some reactions here and makes glutathione over here. And there's cysteine, and there are two enzymes in this pathway. Both of them need B6, vitamin B6. They also need magnesium. And so that's the transsulfuration pathway. And then just for good measure, I've got the rest of the sulfur metabolism here. That comes down below the last one. There's cysteine from the last slide, and it rattles down through all these things, and it makes sulfate and taurine. And then those are used for various purposes, and then some is excreted. So that's kind of the whole sulfur metabolism. Okay, now we're going to start talking about this hypothesis that I've proposed. And I should mention that this hypothesis is very similar to what's been proposed for autism by other people, okay? I mostly borrowed this from autism researchers. And I don't mind saying that, in my opinion, autism and chronic fatigue syndrome are the same disorder. They're the same, okay? Now, I know that if you've ever seen an autistic child and a chronic fatigue syndrome person, you say, well, they're totally different. Well, their symptoms are, but if you look at their biochemistry and their genetics, they aren't so different. And my view is that the differences that you do see in symptoms result from the different ages at onset. If the person gets this disorder when they're 1 year old, the brain is not fully developed, and it affects the development of the brain, and that person will have autism. But if a person is 30 years old, the brain is fully developed by that point. They will get some cognitive problems, some memory problems, and so on, but they won't have the autistic symptoms. They'll have chronic fatigue syndrome. And the symptoms they do have, the child also will have, but you don't hear about them as much because the brain symptoms are so severe in autism, everyone focuses on that. The psychiatrists are the ones who defined it. So, what happened here? Did I push the wrong button? Or? Well, looks good on the screen here. But. So anyway, the adult will have the same gut problems, the same immune system problems. They'll have toxicity buildup. They'll have oxidative stress. The child has all those too, okay? But the adult won't have the autistic brain symptoms. Okay, thank you. Okay, so this is the hypothesis. What causes it? Well, I made the argument before here that there's a genetic predisposition. Yes.
Why isn't cysteine involved in this? Cysteine of methionine.
Why isn't cysteine involved?
Cysteine, yes.
Uh,
Isn't involved in this?
oh, okay, it is involved. Let me back up. Maybe okay, there's cysteine right there. Okay, and yes, it is involved. It's the.
Cysteine.
I'm not hearing you. I guess.
Cysteine. Not cysteine.
Cysteine, I-N-E, cysteine. Oh, okay, yeah. Well, when cysteine becomes oxidized, it's cysteine. Okay, yeah. And that does happen with oxidative stress. You lose a lot of cysteine to form cysteine. So that can, you're right. I mean, that can cause a decrease in availability of cysteine, which can hurt your glutathione production. Yeah. Okay, so the genetics combined with stressors, okay, that's the cause in this hypothesis of chronic fatigue syndrome. And the stressors can be a combination of the ones we talked about earlier. They initially raise cortisol and epinephrine. They deplete glutathione by various direct and indirect mechanisms in a person who has the genetic predisposition. And as I said before, cortisol usually goes down later on, but initially it goes up. And the stressors deplete glutathione in various ways. If you're talking about toxins, they can directly bind to glutathione and take it out of the body. If you're talking about too much exercise, like running marathons, that makes oxidizing free radicals, and glutathione has a role in trying to control those, so that depletes the glutathione. And you can go down the line. If it's epinephrine, if that's at a high level for a long time, it can auto-oxidize and form a quinone, and then you can oscillate back and forth between a quinone and a semiquinone, and that makes free radicals. So there's another load on glutathione. And so you can go down every one of these stressors, and you can find a mechanism for how they demand glutathione. So that's my argument, that you have genetics plus stressors, and they end up depleting glutathione. Now, everyone experiences stressors. Why doesn't everybody get chronic fatigue syndrome? Well, it's genetics, okay? It's my belief that you can take any human being, I don't care if it's Superwoman or Superman, you load them up with enough stress long enough, their health will crack at some point. Now, the way it cracks is going to depend largely on their genetics. They may get a heart attack. They may get cancer. Some of them get chronic fatigue syndrome. Those are the ones that I'm interested in here, okay? So it's just a matter of too much stress is going to do things to a person's health, and what it does depends on genetics. Depends on some other things too, like their history and their nutrition and so on, but genetics is a big part. There hasn't been a complete genome study. Maybe someday there will be, but there is evidence for the genetics, as we talked about. Okay, so now in this hypothesis, glutathione goes down because of the stressors, and the next thing that happens is there's a functional deficiency of vitamin B12. The reason is that glutathione normally protects B12. If it's not there to protect it, then you may still have enough B12 according to a serum B12 test. You may even have more than normal, but your cells are not using it properly, and that's a functional deficiency. And so that's what happens there. And when you have that functional deficiency, now the jobs that B12 is supposed to do don't get done. And one of those is to support methionine synthase. So now methionine synthase becomes partly blocked because there's a functional B12 deficiency. And then sulfur metabolites drain down the transsulfuration pathway because they're not being converted back to methionine because this reaction is blocked. Okay. Well, the next thing that happens is that the folates drain from the cells. And that happens by what's called the methyl trap mechanism. And the idea is that you continue to make methylfolate, but there's no place for it to go because that reaction is blocked. So methylfolate leaks out of the cells into the blood, and the whole folate level in the cells goes down. So the cells now become depleted in folate, as well as having a functional deficiency of B12. Well, then there's a vicious circle that forms between.
Your red dot gets onto your
Yeah, I'm hitting there. Okay.
computer. That's why it doesn't.
All right, I'll try it over here. So there's a vicious circle formed then between the methionine synthase block and the glutathione depletion. And that's what makes it a chronic disease. That vicious circle is not broken from then on unless you do something about it. We're going to talk about what you can do about it. But this is why chronic fatigue syndrome is chronic. That vicious circle locks in the problem of glutathione depletion and the methylation cycle block and the B12 dysfunction. So then I contend that most of the various biochemical abnormalities and symptoms in chronic fatigue syndrome result from these features, okay? Glutathione depletion, functional deficiency of B12, deficit in methylation, and the low intracellular folate. And those are all locked together in this vicious circle. So if you understand that combination, you understand the source of everything in chronic fatigue syndrome. That's my opinion.
The glutathione is the last sort of defense in the cell. And if you increase other antioxidants like superoxide dismutase, catalase, glutathione peroxidase, you help protect glutathione.
They do work mutually, that's right. They support each other. There's a network there. Yeah. But if glutathione is depleted, you really have to do something about the glutathione. The other ones on their own can't do the job that glutathione does.
Where does one measure glutathione, in white cells or where?
Well, I'm going to come to that here, but we measure it in blood plasma. We figure that's the best place, but you can also measure it other places, but that's the one that's the best indicator for what it is in the tissue cells, okay? All right, so what I'm going to do now is elaborate this hypothesis, and what I mean by that is I'm going to try to explain, make the case for how this vicious circle explains everything in chronic fatigue syndrome. And I won't have time to explain everything, but I hope you'll get the idea that it really does explain quite a large number of things, and that's what we're going to talk about. So how do I do that? Whoop. Well, I have kind of two approaches. Now, I came from the world of physics, engineering, and things of that sort, and we use what's called a Gedanken experiment, or a thought experiment. And the idea is you don't actually do an experiment, you do it in your head, okay? And you do it on the basis of having some theory, so you can say, if I did this, what would happen? And you can say, oh, well, the theory would say this would happen, and so on. So you can do that all in your head. So we do Gedanken experiments, and that's sort of the, the forward approach to projecting this theory. And then the other approach is to go backward and say, okay, you have some symptom. What's the physiology of that symptom? And then what would cause that physiology? And you work backwards and see, can you really link it up honestly with this hypothesis? And I claim you can, okay? So that's what we do. So here's the first part of it. If glutathione were depleted, what would you expect to see? This is the Gedanken experiment. Well, you say, well, what does glutathione usually do? What if those things weren't done? What would happen? Okay, and here's what you find. Well, you'd expect to see oxidative stress. Well, we have that big time. 20 papers are saying oxidative stress, okay? So we have that one. Mitochondrial dysfunction and low ATP output. Yes, we have data for that. There's a lab in the UK where Dr. Howard measures that routinely in people, and he finds a lot of dysfunction in the mitochondria. Diastolic dysfunction of the heart, leading to low cardiac output. Yes, that's observed. Paul Cheney runs echocardiograms on all his patients, and he sees diastolic dysfunction. Buildup of toxins, including heavy metals. Yes, big time. We see that in urine tests, hair tests, stool tests. Immune dysfunction. Yes, particularly the cell-mediated immunity. Herpes family viral reactivation. Yes, we see herpes family viruses of all sorts. Thyroid problems. Somebody here likes thyroid. Yes, okay. We see thyroid problems. Okay, this one is a little more involved, but you see lowered secretion and dysregulation of certain cysteine-containing secretory proteins. Now, secretory proteins are proteins that are made inside the cell, just like all proteins are, but these are exported from the cell to do their job somewhere else. So, for example, the peptide hormone family, those are secretory proteins. So they're made in the cell, but they go out to do their job somewhere else in the body, okay? Well, the secretory proteins that have cysteine in them are of particular interest here because glutathione prevents cysteine from being oxidized to cysteine. Who mentioned that here, okay? And that's very important. When you're making a protein, you're hooking together amino acids in a chain. And you want the cysteine to be cysteine, not cysteine when you do that. But if there's not enough glutathione, it oxidizes to cysteine, and now you've got a chain that's not the one you want. And so the quality control system looks at that and it says, this is no good, and it takes that protein apart and recycles it and tries again, and it fails again, and so on. So you just don't get very much of the hormone to go out of the cell because you can't make good molecules without the glutathione. Well, that influences ACTH, which is the HPA axis. Okay, that's the reason when you do a cortisol saliva test, 24-hour, you'll see a lot of them are flat. They're flat. Instead of starting high in the morning and going down like it's supposed to, it'll be low and flat. Well, I claim that's due to the ACTH not being properly made. Antidiuretic hormone is also affected, and that's why the urine volume is so high. I had one guy who was urinating 7 liters a day, okay? The average is 1.5. So getting up all night, urinating, thirsty all the time, drinking a lot of water, that's a problem with antidiuretic hormone. Perforin, this is the molecule that's used by natural killer cells and the CD8 T cells. And this is a molecule that those cells use to poke a hole in an infected cell so they can put in a granzyme, a poison pill, and kill it. Well, they don't make enough perforin. This has been observed in chronic fatigue syndrome. And I suggest the reason why is that perforin is a molecule that normally has 20 cysteine residues in it. It's one of the highest ones. And so that one is very hard to make if you don't have enough glutathione. So that's the argument there. Well, okay, what if there's a functional deficiency in B12? What would you expect to find? Well, high methylmalonate, and that's a test you can run in the urine or in the blood, and sure enough, we see that it's high. Partial methylation cycle block, well, that's observed. Energy deficit, that's observed. Neuropathy, in some cases we do see neuropathy. Macrocytic anemia, not usually observed, but we do see a high mean corpuscular volume in the complete blood counts in a lot of cases. Maybe it's not outside the reference range, but it's high. And that's headed toward a macrocytic anemia, but hasn't quite gotten there. Well, what about if the methylation capacity is diminished? What would you expect to see? Overexpression of genes because of lack of gene silencing. That's what we just talked about. And just before I came here, I got an email from a researcher at the CDC, and they're about to do a complete methylation study of the whole genome in chronic fatigue syndrome. And she heard my talk in Ottawa, or saw my poster, and she's kind of worried that she's going to see, high gene expression across the board on the average because methylation is low. And I said, well, it could be, could be. And so she's going to look for that. If she finds that, there's going to be a lot more interest in this hypothesis at high places, okay? So the Centers for Disease Control has been very influential in chronic fatigue syndrome. They defined it, and they do a lot of the research on it. You would expect to see lowered synthesis of choline and creatine because those both require methylation for their synthesis. What has been observed is an abnormal ratio of those two in magnetic resonance spectroscopy studies of the brain. So we don't have good absolute numbers yet for these, but we know that their ratio is abnormal. And so that suggests that they're affected, or at least one of them is affected. Carnitine, that's another one that requires methylation, and sure enough, that's been reported to be low in chronic fatigue syndrome. CoQ10 also requires methylation, is reported to be low. Now, myelin, we'll be talking more about this later, but what is observed is that a person's processing speed in their brain and also their reaction time is much slower than normal. And that suggests a problem with myelin, because myelin is what allows you to send nerve impulses rapidly down the axon. So if myelin's got a problem, it slows everything down. So that's consistent with a problem in myelin synthesis. Okay, well, what about folate? What would that do? Well, you'd expect to see a rise in FIGLU, and that's observed in urine testing.
Can you go back to the slide before? Before the slide?
Back more?
No, there was a slide before the one you were talking about. Q10 now.
This one?
Yeah.
Uh-huh.
Okay, thank you.
Okay, by the way, if you don't get all this written down, you can certainly have a copy of these slides. You can either contact me or maybe Helen or somebody here, and we'll make sure you get a copy. Okay, back to folate here. You'd expect low red and white blood cell counts. Well, we do see low white cell counts in some cases. And after this kind of treatment I'll be talking about, we have seen white cell counts come up. So that suggests that it is related. Elevated MCV, and we see that, as I mentioned before, in some cases. Hair loss, observed in some cases. Poor digestion and absorption, well, it's observed, but there can be a lot of causes for that, so it's hard to be real specific, but at least it's consistent. I can't say it proves anything, but it's consistent. Well, okay, now, why is there more chronic fatigue syndrome in women than in men, if you believe this hypothesis? Well, here's my argument for that. Women make more estrogens, we all know that, right? And they have to be metabolized. Well, the metabolism of estrogen, if there are certain polymorphisms in the genes, this becomes a problem. And I have an example here that Helen has supplied for me. See, where's the switch here? Oh yeah. Requires a little power, huh? Okay, now. I guess we'll have to kill this other one for a while, huh? Oh, you have a
black.
Oh, okay, blackout. All right.
One question. Could it not be the structural oppression of women in society?
The structure, what?
Oppression of women in society.
Oh, well, that certainly raises the stress on them. I agree with that, yeah, definitely. But I think there's an additional factor here that's more biochemical. And of course, I love biochemistry rather than psychology. But yeah, there's no question about that. I don't know what Swedish society has been like, but in the United States, in the 60s and 70s, there was a kind of a major cultural shift, and the women were encouraged to take on employment outside the home. And that added additional burden to them because they still maintained responsibility for the home and the family, and you know, it was very exhausting for many women. So definitely that's a factor. Okay, this is a report from the Genovations polymorphism study for the detox system. And you notice here that these red spots indicate polymorphisms in these particular cytochrome P450 enzymes. Well, there are 4 of them here, but the one I'm particularly interested in for this issue is 1B1, okay? And then on the next page, the same person has a homozygous, a double polymorphism in COMT. That's catechol-O-methyltransferase, and that's used in the metabolism of estrogen, as well as the metabolism of the catecholamines like dopamine and so on. And then finally, there's also right here, now that happens to be indeterminate in this glutathione transferase. A lot of times it's clearly missing. This one, they're not quite sure, but a lot of times it's missing, or there's a problem in one of these here. So what I have seen in a number of women with chronic fatigue syndrome is this combination of polymorphism: CYP1B1, COMT, and a problem down here in the glutathione transferase. And then sometimes down here too, that's a superoxide dismutase, which you were mentioning contributes to controlling oxidative stress. So the point of all this is that that combination causes, I'm not sure how to shut this off. Pull the plug, I guess, huh? Okay, thank you. Well, that combination causes what's called redox cycling to occur, and that makes more oxidative stress. So my argument is that women who have this combination of polymorphisms, when they're trying to metabolize the estrogens, they have an additional source of oxidative stress that the men do not have, okay? And as I said, the most common age of onset is around 30, which is right in the reproductive age, so I think that's consistent. So, but yeah, stress definitely is another issue. Okay, so then the point is that...
Does that take place also if you add extra estrogen, as we know now that they are dangerous, they give more breast cancer and more hearts?
Yeah, I might, that's right. And I might mention that this model that I've been talking about here with these problems, I didn't invent that. It's a model that's been floating around in the literature quite a while as a possible cause of breast cancer because oxidative stress also impacts DNA, mutates DNA, which then could lead to cancer. So this is a common model for that. Okay, I guess this is kind of what I've been talking about here. Here's estradiol, and there are several pathways for metabolizing it. And normally, it goes largely through these other pathways. But when you have a polymorphism in CYP1B1, this is one of the polymorphisms that increases the activity of the enzyme. Most of them make it go down. They damage the enzyme so it doesn't work as well. This one makes it go faster, okay? So more of the estradiol goes down this pathway, and then if this one is damaged, that one slows it down, okay? So then it can't go this way. So it goes down here, it can't go that way very well, so it heads out here, and here's where it forms the semiquinone, and there's the quinone, and if glutathione was working well, you'd get rid of that. But if it's not, if you've got a polymorphism there too, now you can't go that way. So what happens is it goes back and forth. See these arrows here? And every time it does that, it makes a superoxide ion, which is an oxidizing free radical. So this is redox cycling, and I claim that that's the reason women have more chronic fatigue syndrome than men, all right? Now, this has not been studied rigorously. It's a hypothesis, but I do have at least several cases of that same set of polymorphism. Now, those polymorphisms can occur in men as well, and they do. But it doesn't matter as much because they're not having to deal with estrogen. So there you go. All right, now I want to start talking about the energy metabolism. And this is kind of this is a mitochondrion here that we're looking at. The zigzaggy thing is the inner mitochondrial membrane. And here are the sources of energy. There's glucose, blood sugar, and there's fatty acids. And they enter into the mitochondrion. And this is the pyruvate dehydrogenase complex. So glucose goes through the glycolysis chain, makes pyruvate, and then that's converted to acetyl-CoA, and that goes into the Krebs cycle. And fatty acids, on the other hand, also enter in the same place. They come in at acetyl-CoA here after going through beta oxidation and breaking down the fatty acid into acetyl-CoA. And then the Krebs cycle then generates NADH and FADH2, and they feed the respiratory chain, also called the electron transport chain. And then that produces the energy to, what did I do here? Did I step on something again? Oh, there it is. This makes it interesting. Well, anyway, right here is where ATP is made, okay? So all of this goes into feeding energy to make ATP, converting ADP to ATP. And ATP is the energy currency of the cell used for driving the muscles and various biochemical reactions, the nerves, and so on. Okay, so well, that's how it's supposed to work. And here's a detailed picture of the mitochondrion. And it has this inner mitochondrial membrane here. And so the enzymes that we're interested in are on that membrane, the ones that finally make the ATP. So if that membrane becomes damaged, it lowers the ability of the mitochondrion to make ATP. And so I'm going to argue that, and I think you mentioned this before, mitochondrial dysfunction is the cause of the fatigue in chronic fatigue syndrome. Initially, we have depletion of glutathione, increase in oxidative stress, and then that inhibits enzymes in the Krebs cycle and the respiratory chain and lowers the ATP production. In addition, we have a problem with the methylation cycle, and that lowers the production of carnitine, CoQ10, phosphatidylcholine, and creatine, and all of those are needed by the mitochondria also. So it gets hit in a different place here by the fact that methylation is got a problem.
And then you have a modern treatment with statins that decrease.
Well, I know. That's not something I recommend, especially for these people. That's right. The statin drugs deplete coenzyme Q10, and that can cause problems in the muscles, the heart, other places. Okay, then over time, toxins build up because glutathione's not there to take out the toxins. And some of these toxins end up in the mitochondrion, and they block various enzymes. So as time goes by, more and more problems accumulate and cause more trouble for the mitochondria. Well, then, the fact that ATP production is lowered hinders the energy supply to the ion pumps in the cell membrane and in the mitochondrial membrane. And so now the cell is less able to pump in the minerals it needs and pump out the ones it doesn't want. And so now the essential minerals become depleted and calcium goes too high. So that ends up affecting it as well. And so all of this then adds together like straw on the camel's back, and pretty soon the camel just can't move anymore, okay? And that's glutathione, that's mitochondrial dysfunction. Okay, what about the post-exertional fatigue? What's going on there? Well, we know that exercise causes a higher rate of production of reactive oxygen species. Metabolism always produces these. It's a question of how much and whether you can control them when they're formed. And if you go for more exercise and you don't have glutathione to take care of them, well, you're going to have more oxidizing species, and you're going to damage the membranes of the mitochondria. They're a lipid membrane, and they're very sensitive to oxidation. Well, then, if the mitochondria become too badly damaged, I don't know, maybe my hand is too hot or something. Huh? I'll hold it on the end here. If the mitochondria becomes damaged too much, then it's recycled, okay? It goes into a lysosome, it just dissolves it, recycles all the parts, and uses them over again. That mitochondrion goes away. So then you have to make new ones, and they're made by dividing the ones that you have, okay? So it takes some time for that to happen. And this is something I would like to find out more about. How long a time does that take? If it's about a day, then it's going to give me some more evidence for this hypothesis.
And then you need more thyroxine.
Tyrosine?
Thyroxine.
Oh, thyroxine, yeah, to stimulate that process.
Guyton said that the only role for this enzyme is to stimulate mitochondria to produce more and to make more mitochondria.
Yeah, and to make more enzymes in the mitochondria too. Yeah, that's right. So that's right. The thyroid hormone has a big role to play here. And of course, some of the people are low in thyroid, so that makes it even harder. So that's a very good point. So I think that this explains why too much exercise can be damaging to people who have ME/CFS. And you know, it's a very unfortunate thing that in the UK, the psychiatrists have said, well, one of the most wonderful treatments for chronic fatigue syndrome is exercise, okay? Get out there and graded exercise. And I think it's killing the mitochondria, you know? And there's a website where patients rate all the various treatments that have been proposed, you know? It's sort of an informal poll. And there are something like 150 different treatments and lifestyle changes that are listed there. The one at the very bottom is drink alcohol, and the one just above it is graded exercise therapy. It's the most horrible one, okay, right next to drinking alcohol, which almost kills most of them, you know. So not a good thing, but that's where we are. All right, now I want to talk about the secretory proteins. I mentioned this before, but here's the general mechanism. You know, here's the nucleus, has the DNA, and that has the code for making the RNA. The RNA comes out here, and then it joins to you want to take a break?
Yeah.
Oh, okay. This is a good time to take a break.